Alrighty. Hi there, this is the walkthrough video for Thursday, the 18th. I got it right in try one, week 7. Hope you're doing well. Today I'm going to ask you to compare and contrast active transport, facilitated diffusion, and non-facilitated passive transport. I'm going to ask you what's the same about these types of cell membrane crossings and about what's different. And I'm asking you to use 1 to 3 good sentences. Now, you may or may not have finished the notes on Unit 2. Remember, they are going to be due next week on Tuesday. But even if you haven't, we've been talking about this in class. We've done the membrane structure and function POGL, and you drew a little diagram to help you if you need it. Recall ATP, which is going to be an important part of your answer, is how the cell stores energy, right? And so how it stores energy is like this. So now you have seen the official explanation. When you let go, that delta G can offset the cost of energy requiring reactions, which is how the stuff in our cells get done. So today we're going to talk about how water moves through cell membranes and what the result is, because you're going to be applying this knowledge to next week's labs. So today I have some notes on osmosis, and I have a sheet about, excuse me, I'm kicking the thing I hate under my desk. And I have some notes on water potential. I want to remind you that posters are due tomorrow at the start of class. You are welcome to turn them in early after Panther Plus today or after school. Mrs. Watkins and I will be in here our next door meeting, and you can just drop them by if you are finished. So next week's lab has 3 parts. We are going to do them a little out of order because they require different amounts of prep for me. So the first part of the lab, which we'll probably do on Tuesday, is asking about how cell size and cell shape affect cell function. We're going to be using gel cells of different shapes. The second part of the lab, which we're actually going to do on Monday, is we're going to look at a model cell membrane, a simulation of a cell membrane, and learn about how different substances move across that cell membrane. Last but not least, we're going to make quantitative measurements of water movement using real cells from root vegetables or starches or tubers, probably potatoes if I had to guess. So I wanted to show you what happens with water. So this is a phenomenon called plasmolysis. The plasma in the cells, as this video continues, when you add a very salty solution, this is what happens to plant cells. So again, you can see, what do you think the green things are? Yeah, they're chloroplasts. None of the other things in contrast microscopy have any color, so you're not seeing the nucleus, the endoplasmic reticulum. But you can see the cell wall, and you can see the beginning of the cell membrane with that arrow kind of moving away. And indeed, when you treat the cells with a solution that has a high amount of solute of mixed-in stuff, you get this really pronounced phenomena. So that is not great for cells, right? And you can imagine that if you're a plant who needs water and you are not getting the amount of water that you are evolved to get, some not great things can happen for you. So this is an incredible 4 minutes of biology wrap. I cannot recommend it enough. And I'm excited that you are going to get to see this man do this thing that he loves. I don't know him, but I am very impressed with this science wrap. We're not going to watch it. We can't. So I'm going to teach you a new word. That new word is osmolarity. That's the concentration of a solute expressed as the total number of solute particles per liter. So like the 10.0, I'm not wearing my shirt. I was going to wear my shirt. 6.02 times 10 to the 23rd particles is 1 mole. So we talk about moles per liter. We're talking about osmolarity. Remember, solute is the dissolved stuff in a beaker, and the solvent, in our case, is always water for biology. So with that in mind, I'm going to show you the water notes here. Where are all these things? Great. This is going to go down here. Please hold, please hold. Okay. So I gave people this big notes sheet where I tell people there are 3 types of solutions. There are cartoons of what happens to water when a cell is placed in a solution. And then this is what happens to cells. You saw one of those experiences just now. So these are the notes I'm going to give you. So don't write anything yet. Let me talk through it, and then I'm going to show you each of the stories. So there are 3 types of solutions: hypotonic, isotonic, and hypertonic. You learned that tonicity means dissolved stuff like tonic water. And so that solutes is what that means. You also might have some ideas of what these prefixes mean. Hypo is less or under, like a hypodermic needle. Iso means same, like an isosceles triangle. And hyper means more. This is a terrible stereotype about ADHD, but it might help you remember. So I wanted to be clear, like we think of kids as being like hyperactive in our community when they're running around and screaming. Number one, that's like typical kid behavior. And number two, sometimes ADHD can look like being very focused on something and not paying attention to the outside world. Or it can sort of manifest as being very forgetful. Or it can manifest in not having great sort of spatial sense or reasoning. So this is a quick inclusivity moment for me. And also, our culture still uses this word. You can imagine that this is a lot of activity. So that being said, what happens? Oh yeah, we'll talk about this in a minute. What happens when you put these cells into water? This is all supposed to be the same cell, and it all has the same number of dots of dissolved stuff. This is a solution that has less dissolved stuff than the cell. By the way, we're not counting. It could be salt or sugar on the outside and like proteins and amino acids and tRNAs on the inside. This process, because of the laws of thermodynamics, is just about the amount of dissolved stuff. So this is not a lot of stuff. This is the same concentration of stuff, iso, same, and this is a lot of stuff. So what happens in these cases is that water will flow to the area of less water concentration, even if the stuff that's inside the cell doesn't have a way out. Now, the name of the protein that you'll learn that goes through these things are called aquaporins. And aquaporins are pores in the membrane through which water moves, as you'll remember from the extension questions on one of our POGLs. So water is always moving in and out. Do you see how I wrote this here? Water is always moving in and out. But I've also shown you the net movement of water. The net result is that water moves in this case. The net result is that there's a balance of water in an isotonic solution. And the net result is that water moves out in a hypertonic solution. At the bottom of the screen, I've shown you some of the things that happen. So cells without cell walls end up popping. Cells with cell walls can end up kind of being plump and delicious. This is why the Mr. Mists the carrots at the grocery store, or baby carrots are stored in that bag with a little water in the fridge, as plump as possible. Now, in an isotonic solution, the cell doesn't change its shape or pressure. And so these are the types of solutions you want to give people for IVs, the types of solutions that if you are investigating animal tissues in the lab, you keep those tissues in a suspension that is isotonic. And then hypertonic solutions have their place too. They are how we have salt-cured jerky. They are also how we have made raisins and dried apricots. They are cultural traditions of preserving food, as you'll learn by watching Glenn's video about what happens. Poor Glenn. It's quite a video. Here are some close-ups in case you needed them. There you go. Okay, cool. And then I'm going to ask you to complete the water potential assignment. If you get stuck, you can watch my friend Paul say the exact same things in this video here. And you do not need to complete this because we just did it, right? So we're showing you some images here and telling you some stories about water flow. This is a review of the tonicity video on AP Classroom, but I think it's really important to talk and walk you through this. So you are going to need to know things about pressure potential, about solute potential, about what happens when solute potential is positive and negative. And it helps me to remember that distilled water is 0 bars of water potential and moves up. If this is confusing, watch Paul's video. He's going to explain it for you. And there is a quick problem at the end. You're not turning this in, but it is a really, really useful tool to understand this tricky topic. All right, last but not least, here is a real world question. Let me make it a little bigger for you in the video. Sorry, it's blurry. It's one of the released questions from the College Board. So what is happening here? You are going to pick the correct answer and tell me in one sentence or sentence fragment why it's correct, just applying the learning that we've done today. All right, that's it for me. Project day is tomorrow. Take care out there and see you soon.